Hello, everyone, and welcome to today's presentation. I do apologize for my voice. I recently had nasal surgery. Today, we're going to look at the article Complement Factor H contributes to mortality in humans and mice with bacterial meningitis. It was written by the Dutch scientist Sumirijn Kassenmuntelib in the Journal of Neuroinflammation. My name is Sikander Nielsen. A quick introduction for you guys. So, bacterial meningitis is a bacterial infection of the meninges, which are the three layers that line our brains. And despite vaccines and antibiotic, pneumococcal meningitis is still the most common and lethal form of meningitis. It is caused by the bacteria Streptococcus pneumoniae, which we can see out here to the right. They are a gram-positive diplococcus, which means they come in sets of two, basically two pairs of spheres, as you can see out here to the right. They cause 18 to 37% mortality, and 50% of survivors experience sequelae, which are basically injuries and disabilities after the infection has occurred or after surviving the infection. Serotype replacement by the bacteria itself, as well as antibiotic resistance, are the main reasons for these high mortality rates in bacterial meningitis. Former animal trials have shown that the outcome, we're talking death or survival outcome, has been related to the severity of the inflammation in the subarachnoid space. The subarachnoid space is the space underneath the arachnoid layer. The arachnoid layer is the second layer of the meninges, and this outcome can be improved by modulation of the inflammatory response. Complement factor H. So, a little background info on complement factor H. It is a large glycoprotein found in human plasma. It is encoded by the CFH gene found on chromosome 1. It is a control protein of the complement system. Now, the complement system is part of the immune system. It is not the same as our white blood cells or our natural killer cells or anything like that. However, the complement system does enhance the ability of antibodies, phagocytes, etc. to recognize, bind, and kill pathogens. The complement system is actually a series of smaller proteins that sort of trigger cascades that help fighting off foreign invaders. Now, this image here kind of illustrates this cascade pretty well. At the top of your screen, you will see an invading pathogen, and a human antibody has bound to the yellow antigen on the surface of this invading pathogen. A C1 complement protein has bound to the human antibody, thus triggering this whole complementary cascade. Now, I'm not going to get too into detail with how this whole cascade evolves, but I do want you to notice how the splitting of the C5 complementary protein ensures two things. It ensures that the host cells, the innocent bystanders, are not attacked by the inflammatory response, and it also forms this huge cylindrical complex in the invading pathogen along with C6, C7, C8, and C9 that will eventually ensure that the invading pathogen swells and births, sort of like a cell in a hypotonic solution. Now, factor H is not one of the attackers in this cascade. It's actually not even, if you notice in this picture, it's not even part of this whole diagram. However, factor H ensures that the complement system only attacks pathogens and it protects these host cells. Now, how does it do that? It promotes the splitting of the C3 complementary protein. Now, if we look again at this diagram, the splitting of the C3 complement protein promotes the splitting of the C5 complement protein. And as we see here, the splitting of the C5 protein is what will then bind to a host cell and sort of, mark it as one of the good guys and prevent any damage from happening to the healthy human cell. So complement factor H is not An attacker itself, it is more like, you could call it a director that ensures everything goes according to plan. It's sort of like the quarterback that maps out the entire attack and then lets everyone else do their job. Now, the purpose of this experiment, they did not state a clear hypothesis. However, they had several things going on throughout this article. First of all, they wanted to evaluate the presence of human complementary factor H in cerebral spinal fluid and the immunohistochemistry staining for factor H in brains. They also evaluated the role of factor H in pneumococcal meningitis using mouse models. Here they took knockout mice that did not have factor H and compared them to wild-type mice. And lastly, they also wanted to study if factor H could be used as a treatment, similar to a regular drug. Now we're going to go into the methods first for the cerebrospinal fluid analysis as well as the neuropathology. To evaluate the presence of factor H in the central nervous system, they collected samples of cerebral spinal fluids. They did lumbar punctures on 18 patients with bacterial meningitis, thus collecting the cerebral spinal fluid. In your right corner, you can see exactly how lumbar puncture is performed. You basically stick a needle in between the L3 and L4 vertebrae, and then you will be able to tap cerebral spinal fluid through or from the spinal cord. They also took cerebral spinal fluid from 18 healthy patients, which would serve as the control group. The the factor H levels would then be determined using the ELISA test. Brain tissue from meningitis patients was also collected. Actually, it was only collected from one deceased meningitis patient and also from a control patient who died from a myocardial infarction, or a heart attack as we know it. And in order to stain it, they took anti-human factor H antibodies, introduced them to the sample, and stained them using the horseradish peroxidase, which would yield a brown reaction product. And we'll see what that looks like later in the results section. For the part of the experiment with knockout mice as well as the adjuvant treatment, we first had to obtain some knockout mice, and these knockout mice were mice which did not have the CFH gene, which is similar to humans found on chromosome 1. These mice were obtained from Dr. Pickering at Imperial College in London, so these mice were actually donated in order to save some money. Pneumococcal meningitis was introduced to both the knockout mice and the wild-type mice, and then they studied exactly how well each type of mouse would fare. For the adjuvant treatment, mice with meningitis were treated with adjuvant, and adjuvant means a secondary sort of supplementary treatment, and they were treated with human factor H at 16-hour intervals while also receiving antibiotics. And then obviously you also had a group of mice receiving saline as an adjuvant treatment. Obviously the saline isn't going to do anything, so that's just going to be our control study. Apart from saline, they were also receiving antibiotics. For the results section, in the cerebral spinal fluid, we were able to obtain different levels of factor H. First, we see in the control patients who did not have any meningitis, we saw a median level of 1.12 micrograms per milliliter, ranging between 0.93 and 1.55. And in patients with meningitis, we saw a median level of 11.27 micrograms per milliliter, and these levels were ranging all the way from 6.62 to 15.86. So obviously we are seeing high levels of factor H in patients with a bacterial meningitis infection. In the histological findings, we also saw high expressions of factor H in our meningitis patients and lower expressions in the control cases. So Picture A is from a patient with meningitis. If you see the asterisks here, these are what we call Purkinje cells, and they are all stained brown, which indicated the presence of factor H. And we also see, if you look towards the middle, we actually also see tiny arrows that are pointing towards what is called the granular layer, which also is stained brown. So high presence of factor H there as well. B is our control case, and we only saw factor H staining in the blood vessels, which is to be expected since factor H is found in human plasma, usually nowhere outside of the blood vessels, though. For part 3, this is with the knockout mice. We see the wild type mice who still have human complement factor H present. Well, not human complement factor, but complement factor H present. They survived longer than our knockout mice did. Knockout mice began dying already around 20 hours and did not live longer than 30 hours, where we see wild type mice with factor H present in their systems were able to survive beyond 30 hours. At least some of them were, and all of them definitely lived longer than the knockout mice did. Part 4 was actually pretty interesting. This is for the adjuvant factor H treatment. We actually see here that the mice who were giving human factor H did not live as long as the control mice, which PBS just stands for basically saline. The control mice actually ended up living longer than the mice who were given factor H alongside antibiotics. And now for the discussion section. So we saw that patients with meningitis express increased levels of factor H in their cerebral spinal fluid, which indicates that factor H is activated when a foreign invader is present or detected in the human body. It was expressed in the cerebral spinal fluid because we had an infection of the meninges, which obviously line the brain and spinal cord. So that's why we saw it. We saw high prevalence of it there. Furthermore, our mice who did not have the CFH gene and therefore were unable to express factor H in their body died a lot sooner than the wild type mice. We cannot rule out any genetic differences between the mice because these mice were donated from a third party in order to save some money. However, the findings from this study show that factor H plays a crucial role in fighting off foreign invaders and pathogens, at least for a bacterial meningitis infection. Interestingly enough, we also saw that the adjuvant treatment did not have any positive effects, as the mice who were treated with saline actually fared better than the mice who were treated with human factor H. Now, it is not known whether or not human factor H works differently in mice than it would in humans. Obviously, that would be more interesting to see if we would have more effective results if we were to introduce the treatment to human and not just mice, because obviously the two proteins in the factor H protein in mice is different from factor H protein in humans. That's why it's called human factor H protein. So perhaps it would be more successful in humans. We simply don't know. And that brings me to my last point, which is the relevance in science. The study did show the importance of factor H in meningitis infections, which leads me to think that it is very important in other bacterial infections, perhaps even viral infections. I do not know. It also showed the ineffectiveness of adjuvant factor H treatment, which kind of goes against of everything else that was learned from this study. Perhaps there's a different way of introducing FH into the body. Maybe that's why it didn't work so well, and kind of what I touched on earlier, that human factor H might simply not have worked in mice. So perhaps future studies we could use transgenic mice, which would have a body that works in the exact same way as a human body would, or we can simply, as a means of desperation, try to introduce it to human patients. Although, if it will get introduced as a means of desperation, the outcomes are probably not going to be favorable. Furthermore, I think we should look into study of the complement system. Perhaps there are other parts of the complement system that play roles that are equally important as what factor H does, or maybe even more important, and see what a deficiency in that part of the complement system would do, and how that would inhibit further inflammatory responses as well. And last but not least, these are my references. Thank you so much, guys.